It's an exam question on photosynthesis, and it says figure 2.1 shows an apparatus used to investigate the effect of light intensity, so that's important, on photosynthesis. We're then given a picture of the equipment. So we've got a lamp, we've got some pondweed in a beaker of water with a thermometer. The pondweed is in its own boiling tube of water. We've got a capillary tube that's attached to a plastic tube and a syringe, and we've got a ruler with a scale on it. The question says describe, so we've got to explain how to do it, how the investigation would be carried out, and it's give us figure 2.1, so we need to use figure 2.1. So the plant is carrying out photosynthesis, it's underwater, the product of photosynthesis is oxygen, so we get a mark for saying that the plants give off oxygen, and that's during photosynthesis. So those bubbles are produced and they're going to be trapped here. So the bubbles are trapped. But you can see from the diagram, the bubble is now here. So it's being drawn up from where it's being trapped by the apparatus to here. So what's being used to draw it up? Well, there's a syringe here. You pull this syringe, it's going to suck the water along with any air bubbles along this tubing. So we need to say that the bubble is drawn into the capillary tube by a syringe. That's next to a scale, a ruler, so therefore we can measure the length of bubble. And because this is a tube, we can calculate the volume using pi r squared. Right, we want to measure the amount of the volume of gas produced in a certain length of time. So we need to do this after a certain length of time. So e.g. 10 minutes. And then once we've done it once, we need to repeat so that we can calculate a mean. Once we've got 3 results at this set distance, then because we're looking at effective light intensity, we need to move this lamp either away or towards the pondweed. So we need to say move the lamp away or towards pondweed. And because this is A-level biology, just measuring the distance is really not good enough. We need to measure the light intensity. And light intensity is proportional to distance, it's 1 over d squared. So you could explain that you understand that to the examiner. Right, we've got a water bath and a thermometer. This lamp is going to be giving out heat. We know that temperature is a limiting factor of photosynthesis, so we need to use the thermometer to monitor the temperature. So we need to monitor temperature. And we also want to ensure that this light source is the only light source. So we need to do this in a dark room. The final things we need to talk about is how we're going to make sure this is a fair test. So we need to make sure we're using the same plant each time, that we've got the same CO2 content inside the boiling tube with the pond reed each time. We also want to make sure that none of this gas bubble doesn't escape from any of these joints. So final marking point, we need to Vaseline the joints so that it's airtight. So there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 marking points there. We're after 6. This is a copy of the mark scheme, just so you've got it for your reference.